I think it's time to start. I would like to welcome all of you to our session in reproductive medicine in the 21st era. I am Dr. Taha Abdelmajid, Professor of Urology and Andrology in King Abdulaziz University Hospital and Faculty of Medicine. And I'm glad to chair this session together with my colleague, Dr. Kotman. Today we are very delighted to have well-recognized international speakers from eminent institutions all over the world. They are coming from Saudi Arabia, Germany, and the United States of America. And we have the opportunity to look deeply into the molecular aspects of reproductive medicine, starting from genomic methodologies, going to proteomics and identifying sperm-specific proteins, and finally we will look at the Klinefelter syndrome, the new advances in Klinefelter syndrome, especially infertility issues. Before we introduce our first speaker, we have a small announcement, very short announcement. There is some minor change in the schedule. The last presentation, ovarian hyperstimulation syndrome, is cancelled and is replaced by a presentation by Dr. Aisha Al-Alini. Please, my colleague Dr. Kotman will introduce Dr. Aisha.
Thank you, Dr. Taha. And ladies and gentlemen, we are into the final scientific session of the two-day IGMC Congress. And as Taha introduced, there was an interesting program all along. And this last session is going to be even more exciting. I see a paradox in the arrangement of the program, because this is really a beginning and which foreruns all other programs. This is in fact reproduction, and we have a lineup of speakers, eminent speakers, who will take us through right from embryology and then reproduction in the genomics era and some of the factors that affect reproduction and what would be if something faulty happens in the reproductive process. So with that short brief introduction of the session, let me welcome Dr. Aisha Elani from the CGMR and also CIPM to give us the talk on murine embryology and the effect of GM-CSF. Dr. Aisha, please.
Okay, can I start now? Yeah.
Yes.
I would like first to thank the committee of the conference to give me this great chance to talk about one of my projects, which is going to be about the effect of one of the growth factors on the murine embryo's development, aneuploidy, DNA fragmentation, and morphokinetic parameters analyzed using the embryoscope. We all know that the IVF culture media is actually a hot topic recently, as many companies started to add active groups like growth factors and cytokines to the culture media. And recently, ISHRA set up a group, culture media, to look into the composition and the regulation of making culture media for embryo development. It's well known that growth factors have an impact on embryo differentiation, development, and placental morphogenesis, regulation, and energy requirement. So they are actually important for embryo development. Recently, a new culture media was launched by one of the companies containing 2 nanograms of granulocyte macrophage colony stimulating factor based on its potential in promoting embryo development. It's also known as an adjuster of cell development or blastocyst development. It's important for normal fetal placental development and reducing the rate of apoptosis. While some studies have shown that GM-CSF really enhances the development of the blastocyst, other studies have shown that it has no effect. Some studies have shown that GM-CSF has no effect on the rate of aneuploidy or the chromosomal constitutions in human and murine embryos. A randomized controlled trial was performed to examine the effect of the GM-CSF on the rate of embryo implantation. And they actually found that it can improve the embryo implantation and the survival rate, however, only with the female with history of miscarriages. So we aimed in this study to find out, does really adding the GM-CSF to the cultural media improve the embryo development? And we did our study where we examined actually the developmental potential of the embryos, the chromosomal constitution, the DNA fragmentation, and we added to the study the embryo development timing analyzed using the embryoscope. We know that routinely in the IVF that the embryo selection is made using the morphological assessment, using the light microscope. And recently a document was launched that has all the criteria and the terminology of standard for the morphological grading of the embryos at all stages. However, the time-lapse techniques is a technique based on incorporating a camera into the incubator. So it allows the possibility of continuous morphological observation of the embryos based on images that taken for each individual embryo, together with ensuring the stability of culturing condition by avoiding taking the embryos outside the incubator. It also provides a detailed markers of the embryo development and the cell division. And actually, many studies and publications have really found that there is a relation with the embryo development and the cell division, especially the first stages of the embryo development, from the time of fertilization up to the 5-cell stage. These are some of the publications that actually had been done on human and non-human samples, and they found a really novel markers that correlate the timing of the embryo development together with its potential to develop to the blastocyst stage. So what we did in our study, we did a superovulation of the female mice. After that, we did a mating for female mice with male mice for the fertilization to happen. After that, the female mice were sacrificed and the 2-cell stage embryos were recovered from the oviduct. These embryos were randomly divided into 5 different groups to be cultured in 5 different concentrations of the GM-CSF. These 5 different concentrations were selected based after reviewing relevant studies on the effect of GM-CSF on different species. Together with the control group where no GM-CSF added. These embryos were cultured for 3 days in the embryoscope until they reached to the blastocyst stage. The monitoring of the development of these embryos was happened or performed using the embryoscope based on the images that were taken for each and every single embryos. After that, on day 5, these embryos were divided into 2 groups where they analyzed either for aneuploidy or for DNA fragmentations. So as in our study, we had in vivo fertilization, so it wasn't possible to predict precisely the time of fertilization. So we started our time point from the first cell division that happened. And we calculated the duration of the cell division, so the time the embryos that take to transfer from 2-cell stage to 3-cell stage, and from 3 to 4 and from 4 to 5, rather than taking the specific time point. For example, the embryo at that point became a 2-cell or 3-cell or 4-cell stage. For the aneuploidy screening, we use the FISH technique, and for the DNA fragmentation, we use the tunnel test, which enable the assessment and the quantifications of the nuclei or cells with DNA fragmentation. This is an example for 2 embryos that were monitored using the embryoscope. We can see that we will see at the end of the video that both embryos actually manage to reach the blastocyst stage. So morphologically, we can say that both embryo has a high potential to develop to the blastocyst stage. But if we went back to the start of the movie, you can see that this embryo transferred to the 4-cell stage before that embryo. So the second embryo was slower in its division. And the more time the embryo takes to transfer to the 4-cell stage and the 5-cell stage, the more that expose the embryos to have any errors, either for chromosomal or DNA fragmentation. These two images below, they just show an example on how we analyze the DNA fragmentation. The first image, the cells stained with the DABI to show the number of cells that we have after we fix them on the slide. And those 3 cells that stain with the fluorescent green, those are the cells with the DNA fragmentations, meaning that 3 cells that did not show here has no DNA fragmentation. So, in the result, when we compared our control group to the GM-CSF group, we found that the rate of blastocyst formation actually found to be higher in the control group where no GM-CSF added. And by the time, every time the concentration of GM-CSF increased, the rate of the development decreased. And this could be explained by the negative regulation of the GM-CSF receptor when it presents at high concentrations. When it comes to the aneuploidy rate, we looked at the data from different angles. So first, we have calculated the number of aneuploid or mosaic embryo, and we found that the number was lower in the control group comparing to the GM-CSF groups, although the difference wasn't that significant. And then we looked at the range of the aneuploidy. For example, in the control group, the range was from 0 to 15 or to 10, in the GM-CSF group range from 10 to 30. So we calculated that range and we found it lower again in the control group comparing to the GM-CSF group. The last things we did, we looked at the number of the nuclei or mosaic nuclei in each and every single embryos. So for example, if I have a blastocyst that with 100 or 150 cells, how many of these cells were aneuploid cells? So we calculated the proportion of aneuploid cells in each embryo, and then we calculated the group, and that was again lower at the control group compared to the GM-CSF groups, although it was non-significant. When it comes to the DNA fragmentation, again we followed the same way to look at the rate of DNA fragmentation. So the number of blastocysts with DNA fragmentation, there was no difference between the control group and the GM-CSF group. And the average rate of DNA fragmentation, again, no significant difference. But when it comes to the proportion of the cells or nuclei with DNA fragmentation, it found to be significantly high, significantly different in the 2 nanogram group and the 5 nanogram group. And this could be explained by that the GM-CSF induced the synthesis of anti-PCL2 families in the human neutrophil cells, which therefore reduced the rate of killing the cells with DNA fragmentation. So it reduced the rate of apoptosis by reducing the synthesis of anti-apoptotic PCL2 families. So it allows, in another word, it allows their cells with DNA fragmentation to survive. In another word. The most important point is the time lapse that we looked for. We wanted to know precisely, does really the GMCs affect the development? Does it enhance the cell rate, reduce? Does it make an effect? We actually found no significant difference. We calculated the time the embryos that take to transfer to the 3-cell stage. None of the GMCSF groups showed significant difference comparing to the control, to the 4-cell stage. However, you can see in some of the group, the 2 nanogram group, comparing to the control, it takes actually longer time to transfer to the 4-cell stage comparing to the control group. Even the 5-cell, the T5 as well was longer in the GMCSF group comparing to the control groups. So to conclude, we found that our study showed actually a decrease in the blastocyst development in all the 4 GMCSF concentration compared to the control. No significant difference found in the aneuploidy or mosaicism or the ratio of the aneuploid between the control or the GMCSF groups. However, maybe the high number of aneuploid cells in the GMCSF group could be explained by that. We know that in the cell there is a mechanism that called checkpoints that responsible for holding the occur at the metaphasis stage. It's responsible for initiating the signal that we call it hold, where all the chromosomes align, so can the spindle bind to all the chromosomes before the transition to the next stage, the anaphase, where the 2 chromatid, the sister chromatid, separated. So this action that happened in the cell regulated by interaction of several proteins. And they found that one of the genes called MAD2 genes in the mice is really affected by one of the receptors of BCL2 family, which is part of the GMCSF family. So maybe the disturbance in the GMCSF and the culture media affected the function of that gene, which led to the mis-segregation during the development, which increased the rate of aneuploidy. We saw a significant difference in the mean cell number with the DNA fragmentation between the control and some of the GMCSF group. And finally, no significant difference as we saw in the cleavage time events with the addition of the GMCSF compared to the control. So adding the GMCSF to the culture media actually affected negatively, we can say to some extent, the embryo development. And more studies on larger numbers on the embryos and more randomized control trial are actually required to evaluate the effect of GMCSF on embryo development, rate of pregnancy, and most importantly, the live birth. And embryos and the baby after birth, actually, most important. And thank you. And I would like at the end to thank my PhD team at the University College London, especially my supervisor, Joyce Harper. And I would like to thank also the group at the CGMR and King Abdulaziz University for funding this project, and Unisense company for providing us with the embryoscope to perform this study.
Interesting research. Thank you, Dr. Aisha. The floor is open for questions. Okay, let's move on to the next presentation. Thank you, Dr. Aisha.
Thank you.
Let's move on with the next speaker. The next speaker is Serdar Kaskon. He is the Director of Assisted Reproductive Technology and Senior Clinical Scientist at the King Faisal Specialist Hospital and Research Center. He is also Associate Professor at Al Faisal University in Clinical Embryology and Reproductive Biology. And he has received his MSc and PhD degree from Ohio State University, Columbus, USA. His research interests include PGD, reproductive genetics, in vitro oocyte maturation, and in vitro embryonic development. Let's put our hands together to welcome Dr. Kaskon.
for the introduction, and I'd like to thank the organizer for inviting me to give this talk here. What is reproductive medicine? Let's start with that one. Reproductive medicine is the branch of medicine that deals with the diagnosis and management of the reproductive problems, including having a healthy child. What is genomics? I don't think I need to describe genomics here because it's a genomics meeting. I'm probably the last one to describe the genomics, but in short, it's a study of organism entire genomes, including genes and their functions. How genomics and reproductive medicine comes together, as in any part of the medical field, as the genomics developed, we are adopting what are new to our current diagnostic or treatment methodologies, and there are so many fields, and I choose 4 different areas which we are studying, and each of these could be a topic of a lecture, but I'm going to give some synopsis of what are these: pre-implantation genetic diagnosis, prenatal diagnosis, identifying the genetic basis of reproductive disorders, and a little bit about the functional genomics in the wake of what we are doing in King Faisal Specialist Hospital. Let's start with PGD, pre-implantation genetic diagnosis. In short, PGD, it allows the determination of embryo genotype before implantation. So disease-free embryos can be transferred. How PGD is done for the people who doesn't know? And first, the couple comes complaining from having the affected child for certain diseases, cystic fibrosis, thalassemia, sickle cell, whatever. There are thousands of different conditions. And the wife are induced with hormonal regimen to induce multiple eggs, because in the regular cycle only single oocyte produced, and we need more eggs to do the diagnosis. And in vitro fertilization process is done, and fertilization is achieved by intracytoplasmic sperm injections, and embryos are produced. Then samples are taken from the embryo. These samples can be taken at the polar body stage, but this is rarely done nowadays. Mostly day 3 8-cell embryo, or nowadays it's shifting towards more blastocyst biopsy. Once the sample is taken and it's analyzed for the genetic whether it's a normal or affected, and normal embryos transferred for mother to give birth to healthy babies. And when we look at the history of PGD, especially in terms of the technical development, first PGD was performed in 1967 on rabbit embryos by sexing. In this case, it was a simple nuclear staining. Of course, at that time, probably it was not simple, by looking at the bar bodies. Bar bodies are the inactivated X chromosome. That's why it's only seen in the female embryos, and it's not present in male embryos. And it was done in rabbit. For first PGD baby was born in 1990 for X-linked disorders. For this to happen, we need to have the first IVF baby in 1978 and the introduction of PCR in 1985. Then in 1992, again, first baby for PGD for the single gene disorder was born for cystic fibrosis. In this thing, the regular gel electrophoresis is used, and at the beginning it was successful, but there were some misdiagnoses. Within first 15-20 birth, there were 2-3 affected babies. That's why new technologies has to be adopted. Then fish replaced looking at just Y chromosome by gel electrophoresis, because you are looking for both X and Y chromosome, and also fluorescent PCR, haplotyping, and CGH was introduced between 1996 and 2000. As mentioned in the morning with the human genome projects, lots of new technologies emerged, and these are adapted into PGD. And among those, it's like carrier mapping, array CGH, and next generation sequencing. What led to these technologies to adapted to PGD was whole genome amplification. And basically in PGD, we have very limited amount of DNA, sometimes single cells, sometimes very few cells. And you cannot do all these genomic studies, and whole genome amplification need to be applied. We are one of the first to apply whole genome amplification to PGD field, and We published the proof of principle in 2004 and clinical validation in 2005. And in 2007, we said that in a review, it will be a new era for pre-implantation genetic diagnosis. In fact, it was. And basically, single cell is taken, putting into a PCR tube and lyes, and whole genome amplification, we are applying multiple displacement amplification. In the regular PGD, you only use this tube when you do for single primer, but in this case, microgram amount of DNAs are produced and many tests can be done at the same time. Basically, you have the single cell. Now with the whole genome amplification, you can do STR analysis, next generation sequencing, carrier mapping, HLA haplotyping, array CGS, SNP arrays, anything available to genomic DNA can be applied to also PGD. Basically, this way in the classical PGD, by looking only the certain area of the gene, now we can look at the whole chromosome or whole cell genome in the single experiment. I will tell you about a little bit about carrier mapping, and carrier mapping is utilizing the SNP genotyping, and it allows the differentiate disease associated haplotypes. In this case, we need to have the reference child, whether normal or affected, and from this one we identify which haplotype from father is affected, which haplotype from the mother is affected, and then when embryo tested, we look for this affected haplotype, and those embryos can be excluded from transfer, and these embryos will be considered as carrier, and this embryo will be normal. It's a long procedure, but this is basically a summary of the things. And we were, again, part of the international validation of the carrier mapping, and 44 PGD cycles from 25 different single gene disorders were tested, and total of 218 embryo used, and carrier mapping was 98% concordant with the classical method, and it gives much more information than the regular PGD. And subject to SNP coverages, it reduces or eliminates need for direct mutation detection, because it's going with the haplotyping. Once you know the disease haplotype, without looking at the mutation, embryo can be chosen by this method. And there was one recent publication came in September this year, and tens of live births following the adoption of carrier mapping was published. And one of the beauty of the carrier mapping is you don't only look at this disease-associated haplotypes, also you can look at 24 chromosome aneuploidies. Like in this case, in the embryo, chromosome 9 trisomy is detected, and here also chromosome 6 aneuploidy detected. Not only diagnosing the disease, also the aneuploidies can be detected by using carrier mapping. Also in PGD area, what genomics brings is the probably embryo selection. It is well known that as the age of the woman goes up and the embryo aneuploidies also goes up. When she reaches around 42, it reaches around 80% embryo will be abnormal. And when IVF is done, when there are many embryos, they will be more or less sometimes similar quality, and currently we have only means to look at morphologically under microscope which embryo will be better, to choose which one to transfer, and the genetics of the embryo can be studied. In early 2000, fish was employed to do this, but fish is only fluorescent in situ hybridization. It's only looking at certain number of chromosomes. Also in 2006, 7, 8, around that time, there were several prospective randomized studies showing that fish technique is not good for selecting embryos. It doesn't increase the pregnancy. Even in one study, it was harmful to embryo selection. Then with the genomic technologies, array CGH and the next generation sequencing technologies adapted to embryo selection. And I'm not going to get into the details of the studies, or the technologies, because we don't have enough time. But this is a just published paper looking at the meta-analysis, and when the chromosome analysis by using either array CGH or the next generation sequencing, there are cohort studies and randomized controlled trials. And basically, when we look at the live birth rates from randomized controlled trials, it's almost increasing the live birth rate by 25%. And also, in the randomized controlled trials, the miscarriage rate is reduced by half. In cohort studies show even more effect, but of course we are going with the randomized controlled trials. And currently there are two big studies, randomized controlled trials going, one in Europe and America, another one in China. The result of those will show exactly whether we'll be able to use these technologies in IVF to select embryos. And similarly, next generation sequencing, the beauty of next generation sequencing, again, it can look for the chromosomal abnormalities. Also, it can look for the sequence of the mutation, and this will give direct benefit. Recently, it was also studied in embryos and PGD, and in the embryo, results of next generation sequences was very similar to classical methods. By this way, instead of looking on fishing only one fish, then we can get the whole information from single experiment. And also, the genomic technologies are making difference in prenatal diagnosis. The classical prenatal diagnosis is done after 10 weeks by either chorionic villus biopsy or after 14 weeks by amniocentesis. Both procedures are invasive, and it's at the late stage of the pregnancy, later stage of the pregnancy. It's not very early for some people to consider abortion. But now there is a lot of studies on the cell-free DNA, and this is highly studied in the cancer area, and also it has been studied in the pregnant woman, and there are DNA, free DNA circulating in the maternal blood, and there are DNA coming from fetus from the placental cells. And these cells can be looked or studied non-invasively to see whether baby is having any abnormalities. And during last 3-4 years, there are lots of improvement on this area, and many companies came commercially to introduce this technique to study especially aneuploid of 18 and 21 chromosome. They are using the parallel sequencing and targeted sequencing, and also the SNP arrays in some other companies. And it has been recently validated with using thousands of samples, and it has almost 100% specificity and sensitivity compared to the classical methods. And that's why this is now almost taking over the invasive prenatal diagnosis technique. However, even this new technique, they are still working on around week 10, after week 10, because the sensitivity of the test is better when the percentage of the cell-free DNA, fetal DNA is high. And when it is around 3%, which will be around 45 weeks or less than 10 weeks, sensitivity will go down. To overcome this, we adopted one technology called beaming, and we published this May this year. And basically, we utilize the emulsion PCR. In emulsion PCR, you are doing bubbles, oil bubbles, and inside oil bubbles, you are creating an environment with a single copy of the DNA and with the other primers and whatever, and there will be one bead inside. And amplification will be done, and in this case we use amylogenin gene. Amylogenin genes with single primer, there is a difference in size between X and Y, and we after doing this, you remove the bubble and you stain with different dye for the X and Y, and we run the flow cytometry. As you see, this is a nulliparous woman, meaning that she never get pregnant, and there is almost no Y chromosome. And this is a patient, delivered girl at 4 weeks. That is again almost similar, 0.02% similar to the nulliparous woman. But at the boy delivered at 4 weeks and 7 weeks, it's increasing from 0.75 to 1.3%. We did this on around 20 patients, and this is the average of the nulliparous woman, and these are the girl deliveries. This is at 4 weeks. Basically, when I say 4 weeks, this is the 4 weeks of gestation, and it is 2 weeks after embryo transfer. It's the 2 weeks embryonic time, and this is the earliest time to detect pregnancy anyhow. And in the boy pregnancies, it's very high, significantly higher than these two. And this shows that as a proof of principle, we did not do this as a clinical study. Even at 4 weeks with highly sensitive techniques, the gender of baby or diseases can be detected. And again, this is the comparing 47 weeks of pregnancy in the girl deliveries versus boy deliveries. And the girl deliveries, there were no difference, but the boy deliveries, amount of the Y chromosome is increasing. And the genomic technologies can be utilized identifying the genetic basis of the reproductive disorders. And there are so many studies and so many mutations are being found, but I will be going over something we faced last year. We have a patient, she is 30 years female with 6 years infertility, and she has bilateral tubal blockage, and which is, I mean, in the IVF sense, it's good that it's you consider her as tubal factor, and her FSH was normal, and the antral follicle count is high, and husband is 30 years with normal semen parameters. You expect that when you do IVF, you'll have embryos, and within few cycles, hopefully she will get pregnant. And when she was referred to us from another center, they had 4 XC cycles, and each cycle 11 to 23 oocytes were retrieved, and no fertilization and no ET. And when we did the first IVF cycle, we employed XC because we had only 4 oocytes. In this case, we usually do split XCs, but because of the number of, if it is less than 6, and again, no fertilization, no ET. In the second cycle, stimulation was changed, and we get a big number of oocytes, and 12 were allocated to XC, and 6 with oocyte activation with calcium ionophore, and again, no fertilization, neither in the with calcium, without calcium. However, in IVF, out of 19, 3 fertilized, but only 1 divided with very poor embryo quality, and we still transfer, and there were no pregnancy. And it was an interesting case for us, and I checked our database to find whether there are similar patients. I found 6 patients with the similar clinical outcome, and it happened to be one was her sister and another one was her cousin. We were lucky because when it's a family, it's easier to go for genomic studies. And for these 6 couple, genome-wide SNP genotyping, whole exome sequencing, and all the population frequency, and it was studied. And when doing all this, at the end we come to one single gene, which is TLE6. It's a member of subcortical maternal complex. And there was a mutation converting serine to tyrosine, which was phosphorylation site. Again, in the lymphocyte of the patient, we showed that phosphorylation was significantly reduced in the patient. And we look at the literature, and in 2014, this paper was published in Nature Communication with this TLE6 knockout mice. And when they have the male as the knockout, it's having the child, but the knockout females are completely sterile. And they look at the in vitro fertilization, and this is the controls, which is the normal development, and this is the TLE6 mutant female mice, and the embryos are not developing beyond 2-4 cell, and they are highly fragmented, which was very similar to our study. And the male are fertile, and the females are infertile. And when we did the extended family study, when there are two sisters, they both have the mutation, they don't have any child, they are infertile, but they have brother, again it's an affected brother, he is having 5 children, which is very similar to what was seen in the mouse. By this way, by using all these genomic technologies, unexplained tubal infertility explained, and this paper was published beginning of November. Just briefly, with one slide, the transcriptional profiling also can be studied, and there are lots of studies on this issue by looking at the cells surrounding the oocyte during development, and also looking at the transcriptional profiling and the endometrium, because embryo eventually will be placed here and to be implanted. And the studies are ongoing on this one, the cumulus cell, I think it was Dr. Asid is working on this subject, I'm not going to mention anything, and endometrium, there is endometrial receptivity array, it's again multinational study ongoing, the result, we are waiting for the result, whether it will bring anything. On this level, we did 2 different studies. We look at the transcriptional profiling of granular cells from a patient with recurrent empty follicle. Empty follicle means that there is no oocyte after stimulation, normal stimulation. And what we found on this paper was the apoptotic genes are highly upregulated in the empty follicle syndrome. Again, just recently we published a paper on the gene expression profile of granular cell by looking at the different dose of HCGs. And in this study also, in IVF regularly 10,000 HCG was given. And we look at 5 and 10,000, there was no difference on the gene expression profile, which goes with the other studies showing that as low as 3,500 IU will be affected. In conclusion, by using genomics, now with the PGD, whole genome of the embryo can now be studied. And in prenatal diagnosis, early and non-invasive prenatal diagnosis can be implemented. And the unexplained reproductive disorder can be explained by genomics. And again, in functional genomic side, gene expression analysis might be useful in understanding the follicular drug response and prognosis. Thank you very much. And these are our collaborators.
Thank you, Dr. Serdar. Very interesting presentation. We have time only for 2 questions. Okay. I have a question, Dr. Serdar. How accurate is the methodologies you are using for PGD to detect genetic abnormalities? Or in other words, how much sensitive and specific?
In terms of cardiometric?
Yes, that might lead to wrong clinical decision.
Yeah, cardiomapping, if you are asking specifically for cardiomapping, you know there are technologies which ended up with the misdiagnosis. We know that, but the cardiomapping is a new technology using the SNP arrays, and as I showed you in our study, it's 44 cycles for 25 disease, and the correlation was 98% with the traditional technique.
Thank you.
Thank you, Sardar, for this nice presentation. The question is regarding the single-cell whole genome application. Did you perform any prior optimization to select between platform, the MDA or other techniques, WGA for sample size. And we know that there is some bias when we try to...
Yeah, there are lots of problems with whole genome amplification, but our log in PGD, we are looking for something new. We are not looking for something new to be diagnosed. We know that parents will have this, and MD is supposed to have either this and that. We did lots of studies on this, even the different primers and things like this. For certain techniques, for example, for cardio mapping, it's working very well, and allele dropout rate is around 25%. But especially in the cardio mapping, since there are hundreds of thousands of SNPs, even few of them, or even 1/4 of them are dropped out, you are still able to get the results. In the RACDH, this MDA doesn't work as well. That's why other techniques are being used for different platforms.
Thank you, Dr. Cesar.
Okay, thank you.
Okay, Dr. Serdar tackled the problem of unexplained infertility in the female, but still we have the unexplained infertility of male untouched. We leave this for the Professor Ashok Agarwal, the director of the Center of Reproductive Medicine in the Cleveland Clinic in the United States of America. Professor Agarwal is a well-known researcher and scientist all over the world. He enriched our medical literature with hundreds of high-quality publications. Here, Professor Agarwal today will touch the concepts of proteomics and which are techniques to identify sperm with specific proteins, and will provide us with will elaborate on the future of proteomics in male infertility. Please welcome Professor Agarwal.
Good afternoon, everyone here. First of all, I want to thank the organizing committee of the CGMR for their kind invitation. It has been a great pleasure for me to be here amongst all my friends and colleagues. We have been working for the last 3 years now, and this is my third or fourth visit, and this is the third meeting of the IGNC, so I'm very pleased to be here. I also want to take this moment to thank the chairperson, Professor Taha, as well as Dr. Gautam for chairing this session. So, what I wanted to do today was to talk about some of the research that we have been conducting in our center at Cleveland Clinic. And so my talk is going to be more about the work that we are conducting and just give you some of the latest findings, including some of the research that we have with our colleagues here at CGMR. So the things that we are going to cover are, so the learning objectives will be to talk about very briefly what are the limitations of semen analysis, discuss some of the new advances to evaluate the fertilizing capacity of the spermatozoa, discuss the proteomics and its role in infertility, some of our studies at our center on proteomics and male infertility, what are the challenges of conducting proteomics research with respect to male fertility, the limitations of introducing proteomics for male infertility patients as a diagnostic tool, and conclusions and some of the take-home messages. So this diagram is essentially showing the cellular changes which occur during the spermatogenesis or sperm production, and during the phase of spermatogonia to elongating spermatids are stages where transcription and repair occur before the sperm are released into the epidermis, and there are no more changes that occur once the sperm are released in the epidermis, and it is transcriptionally inactive or silent. So the incidence of male infertility is about 15% of all couples in reproductive age in the United States. And male factor contributes to about 50% of all cases of infertility. And in some parts of the world, especially in the Middle East, this could be as much as about 60 to 70% of the cases in a study that we published recently. In addition, about 25% of cases of male factor account for no identifiable cause and are called as unexplained infertility. So there is a limited amount of information. Semen analysis is one of the first tests that is used to evaluate a patient coming for male infertility workup. As a physician, Dr. Taha will tell that they will recommend 2 semen analysis 2 to 4 weeks apart. But it provides a limited amount of information because it only tells us about certain parameters that can be measured microscopically, such as count, motility, and morphology. But this certainly, I think, does not tell us much about the functional competence of the spermatozoa. So that is a major limitations of semen analysis. There are a number of advanced functional tests. Some of them are described here, many more are not. These are in no particular order: acrosome reaction test, a test for sperm capacitation, sperm membrane integrity, measurement of free radicals or reactive oxygen species, measurement of antioxidant capacity, measurement of sperm DNA fragmentation, lipid peroxidation assay. Some of these tests are being utilized by us in the clinical clinic for routine testing after the semen analysis testing. So those are tests that can be used as an advanced test. So now talking about the proteomics, essentially the steps involved here are in the fragmentation of proteins into peptides, digestion of peptides and LCMS analysis, selection of proteins and by collision-induced dispersion, and selection by mass spectroscopy. So those are the things which involves proteomics. So the current focus is to understand the underlying molecular mechanism of sperm dysfunction. It is important to understand that because that could serve as a potential biomarkers in elucidating the causes of male infertility. So why proteomics? Because the study of cellular components at the molecular level, it helps identify the differential expression of proteins which are either over or underexpressed or are present as a unique protein in a cohort of infertile men with various types of clinical diagnosis or fertile men. Sperm-specific proteins which help identify various metabolic pathways, processes or networks that are essential for the diagnosis of male infertility. And it also provides a greater understanding of protein function. Proteomics allows the identification of sperm-specific protein interaction associated with important steps in sperm metabolism. And for that, sperm is an ideal source because it's a fully functional, mature cell, translationally and transcriptionally inactive. And that's why proteomics is a better omics for studying this, unlike genomics or metabolomics. And it utilizes proteomic tools and bioinformatic analysis. The challenges in male infertility research is that advantages of using proteomics over routine semen analysis is that it confirms the qualitative and quantitative measures of a protein within the spermatozoa. However, identifying ideal biomarker for clinical diagnosis in a complex scenario to distinguish the pathophysiology is only one of the main challenges. Further, in a clinical proteomic study, one sample per individual will only give a snapshot of of the disease. So it is really important that you need several samples to conduct a more thorough understanding of the proteins that are involved. So in longitudinal sampling, where samples are collected over a course of time to determine how the proteome changes over time within an individual, is necessary to identify putative biomarkers. In a study that we published recently, we conducted a comparative analysis of protein in fertile and infertile men, and we ran a 2D gel and conducted Marley-Toff. We found that we identified 4 unique proteins in the sperm of fertile and infertile men, which were seminal gelin 2 precursor, prolactin-inducing proteins, cluster isoform, and prostate-specific antigen isoform. So this was the first study. And then in a second study that we published on 2-dimensional differential in gel electrophoresis, in this study we examined semen samples from 20 normal men with normal semen quality and 32 male infertility patients, divided them into ROS positive and ROS negative by conducting estimation of their seminal free radical content, and pooled sperm were used to prepare for 2DG analysis, and sperm were labeled with fluorescent dyes and excised protein spots, and we then ran the LCMS analysis. The main findings of this study was there were about 1,200 spots in patients with ROS positive and about 1,300 spots in ROS negative, and there were 31 of them were differentially expressed. Of these, 6 spot decrease and 25 spots increase in ROS negative samples compared with ROS positive. There were significant proteins with antioxidant properties seen in ROS negative sample, and this we concluded may explain the significance of ROS in the pathology of male infertility. This is the study that we published. Another study that we published was looking at the proteomic analysis of human sperm proteins with oxidative stress, which is an area of great interest to our program. In this study, we examined semen samples from 53 subjects, 20 donors, normal subjects, and 32 patients with male infertility. And here we again conducted the study, and we found that in this figure shows the percentage of proteins involved in various biological processes such as metabolic process, protein folding, protein regulation, reproduction in response to stress. And the dark blue bars here represent overexpressed protein, and orange bar the underexpressed protein in the ROS positive group. And this figure, it shows the cyclic AMP response element modulator of cramp signaling. Here the spermatozoa circled as 1 in red, and RODF1 and ACE1 are overexpressed, where those in blue, which is LDSC, is underexpressed in cramp pathway in ROS positive group compared to the ROS negative. So there were 74 proteins identified, and about 10 of them were overexpressed and 5 were underexpressed, and the number of proteins that we saw are listed here. The results suggest that these proteins can serve as a potential biomarkers of oxidative stress. This is the article that we published. And this is another study that we published where we looked at the proteomic analysis of seminal fluid from men exhibiting oxidative stress. And in this study, we examined the seminal plasma proteome in men with oxidative stress. The study population was the same. We looked at the seminal fluid, unlike the sperm proteins in this one. And we identified 124 proteins. 7 were common and 7 were unique proteins. And we identified proteins that protect against the oxidative stress. Men with oxidative stress have proteins that are underexpressed or oxidatively modified and contribute to male infertility. So in this diagram, we can see this figure shows the comparative analysis of differentially expressed proteins that were present in the ROS positive blue and and R was negative green bar, or were common to the 2 bars, which are orange. So the differential expression of these proteins in the cellular distribution are shown here in the biological processes in this particular segment, and in the third column are the molecular functions according to the differentially expressed proteins. So the main findings was that men exhibiting high levels of reactive oxygen species show proteins that are either down-regulated or oxidatively modified, and we identify KLK3 as a key protein involved in the majority of pathways, processes, and networks. This is the article that was published. There's another study that was published which looked at the differential expression, differential proteomic profiling of sperm proteins in infertile men with unilateral or bilateral varicocele. And the main findings of this study were that we identified 253 proteins, 64 were uniquely expressed in the bilateral group and 31 in the unilateral varicocele group. Majority of these were involved in metabolism, apoptosis, and signal transduction. Core function proteins were post-translational modification, protein folding, free radical scavenging, etc. And the top pathways were protein ubiquitination and mitochondrial dysfunction. Again, our results provide insight into the underlying mechanism of varicocele-associated infertility in infertile men with unilateral or bilateral varicocele. This is another study which is in the press right now on the proteomic signatures of infertile men with clinical varicocele and their validation studies reveal mitochondrial dysfunction leading to infertility. Here we examine the protein identified from our varicocele studies on unilateral, bilateral, and comparison between unilateral and bilateral varicocele groups. A total of 50 subjects were enrolled, and what we found here is, this is the Venn diagram here, which shows a total of 1250 proteins, 138 were unique to fertile control group, and 140 of them were unique to unilateral varicocele. 120 were unique to bilateral varicocele group. And the Western blot analysis was done to validate our results. We selected 5 proteins for validation, and these were based on their role in stress response and regulation of structural and functional integrity. And these were the down-regulated under-expressed protein were PKAR1, which is cyclic AMP-dependent protein kinase protein, AK7 protein adenylate kinase, CCT6B, which is the T-complex protein 1, and heat shock protein. Upregulated or overexpressed protein were ODF2 or outer dense fiber protein 2. So the overall summary of various studies that we discussed and many more that have been published recently from our program shows that sperm proteomics is an important tool in understanding the basic underlying mechanism of male infertility. Various type of technique such as 1D, 2D or 2DG can be used prior to LCMS analysis, and proteomic analysis and bioinformatic tools are important in identifying the differentially expressed proteins. The differentially expressed protein alteration is responsible for various reproductive functions which are related to male infertility, and proteins of interest can be examined further as potential biomarkers of male infertility. So, the future of proteomic study in terms of the goal is to fully characterize the human sperm proteomes. This is a big job which will take some time, but there are thousands and thousands of proteins. Understand differences between various stages of spermatozoa, identify post-translational modification that will lead to suboptimal sperm quality, develop novel diagnostic tools and therapeutic drugs to improve sperm dysfunction, apply sperm proteomics not only as a research method but also as a clinical diagnostic tool in the field of male infertility. These are my colleagues from Cleveland Clinic, Dr. Rakesh Sharma, Damayanti from now in Kuala Lumpur, Malaysia, Ahmed Ayaz from Istanbul, Luna Samanta from India, Ricardo Bertola from Sao Paulo, Gulfam, Ahmed from Pakistan, and from CGMR, Dr. Murad Asidi, Mohammed Abu Al-Magad, Dr. Adil, and Rola Turkey. And these are some of the studies which were published. Some of them are listed here, some are not, which were in collaboration with our colleagues from CGMR. We are also looking for a candidate to pursue this research in our program. If anybody is interested, they can reach out to me, and we'll be happy to talk about that. And these are some of my colleagues in the Cleveland Clinic. Thank you very much.
Thank you very much, Professor Agarwal. Very stimulating presentation. The floor is open for questions and comments.
We are quite far. I think it's a very promising area. But the main challenges of proteomics for diagnostic purposes in infertility cases is essentially, it is a very sophisticated technique. It requires a great amount of expertise, expensive instrumentation, and it is it is a very labor-oriented technology. The validation is very important because the results are not easily reproducible. It requires immense amount of standardization to achieve something which can be repeated by the same investigators or by other investigators. So I think it definitely is a technique which is very promising as something that may happen, but I believe for another maybe 5 or 10 years, maybe sooner, but it doesn't seem like we have a lot to discover, especially in the spermatozoa proteomics field. We have just started, so it's basically like just the beginning of the whole unraveling of the whole sperm proteomes. There is a lot of work to be done.
Ashok, thank you very much for the talk. Doing the proteomic profiling during spermatogenesis is very important. Like doing it for the spermatogonia, primary spermatocytes, secondary and spermatids. How you would dissect that?
How do you
dissect at the cellular level?
Really, we are not, except for some of the studies where we did separate out the mature and immature sperm using the double density gradient, actually 3-layer density gradient, where we were able to separate the spermatozoa which are mature versus immature spermatozoa or white blood cells. There isn't an easy method to separate these cells in enough quantity for proteomic analysis, because you need enough amount of proteins. So separation is just one part of the puzzle, but the biggest problem is to have enough number of cells then to use it for downstream processes that you require enough number of proteins, not only to run the proteomics but also to do the validation studies on the same samples later on. So it's very difficult.
Dissecting like mature and immature, quite general, isn't it?
Yeah.
My question is, are you going to dissect it stage by stage, or that will be really difficult?
It will be, at least in humans, is very difficult. I think there isn't any such type of technology right now which will allow us to do it in a way which will allow enough amount of cells. You might be able to do it, but the number of cells will be too small, and then it may not be very pure. It will not be very pure population of those cells. So the purity of the cell population will be compromised.
Any more questions? Okay.
Thank you, Ashok. Very nice talk. As usual, my question is regarding the intra-patient variation and intra-group variation between these days, especially that the number of samples is limiting and the amount of as well.
Yeah, those are genuine concerns, and there is a way that you can get around these things, and I think, you know, there are biological variations as well as sample variations and technical variations that have to be taken into account. So for some of the things you can run the samples in triplicate to avoid the variation, especially the technical variation. For biological variations, you can pool the samples and put them all together in a group of samples so that you can reduce the variability as well. So those type of issues have been studied and reported in the literature quite well now. And I think most of the people who are working in the proteomics field are able to conduct these studies quite well.
One last question.
Okay.
My question concerns ROS. Is it the cause of some genetic defects in the patients, or is the effect?
Well, it's the chicken and egg type of situation, which came first. We know that there is a strong association. I mean, I think many of you are geneticists here, so you might be able to tell more. But we know that that reactive oxygen species, or ROS, is an underlying cause of sperm dysfunction through the process of lipid peroxidation. So it causes cell membrane breakdown and causes a variety of damage leading up to the cell DNA damage. So those type of changes are already known and have been published in hundreds of articles, but its direct relationship to genetic changes further than DNA are not studied in experimental setting. I think most of those studies are, association studies that have been published. So I really don't have a clear answer in terms of the impact on genetic.
Okay. I have a question, Professor Agarwal. If you find a defective protein, if there is any chance to fix or rectify this defective protein using proteomic techniques?
That is hypothetical at this time. I mean, we are at a very basic stage of mapping out all the proteins under different clinical, I think, diagnosis of male infertility, such as varicocele or azoospermia. And so, certainly, I think this is a good question because as a clinician, you would like to know. We will probably think that it is possible if we can correct the underlying cause